This conference will now be recorded.
And gametocyte of Plasmodium vivax. Don't write. All of you concentrate here. Don't write. Okay. First one is Plasmodium falciparum tropozoid ring stage. Okay. This is the stage of early tropozoid stage known as ring form. It is angular or signet ring appearance containing a central vacuole and peripheral thin rim of cytoplasm. Okay, look like a ring stage. That's why ring. Okay, next, these species Plasmodium falciparum can able to infect all ages of erythrocytes, mean including old and young stages of erythrocytes. And this ring form occupies 1/6 of RBC. Okay, important for Viva. In case of Plasmodium falciparum tropozoid, what happened? This ring form occupies 1/6 of RBC. Okay, this is the first sexual form that can be demonstrated in the peripheral blood. Okay, for this falciparum, pre-patent period is 5 days. What is pre-patent period? Anybody? After infection, appearance of this gametocytes in the peripheral blood is 5 days. Next, see the ring stage of ring form of Plasmodium falciparum. And another important point here is this falciparum species produce multiple rings in one RBC. Inside one RBC, we can see multiple rings. This is possible only in Plasmodium falciparum species. Okay. This is the picture of Giemsa stain. Okay, here specimen is peripheral blood. Next, gametocytes of falciparum. Okay, this is the gametocyte is the sexual form of Plasmodium falciparum. So this gametocyte development takes place in the blood vessels, spleen, bone marrow, and but only the mature gametocytes appears in the peripheral blood. Okay, development takes place in the internal organs and blood vessels. Internal organs like spleen and bone marrow, but mature gametocytes appear in the peripheral blood. Okay, only the mature gametocytes appears in the peripheral blood. These mature gametocytes either in falciparum or in vivax. These are the infective form, means able to produce infection in mosquito. Here mosquito is definitive host or intermediate host? Definitive host. Okay, because sexual cycle takes place in mosquito only. Here intermediate host is man. Next, the gametocytes of falciparum are crescent or banana shaped. Very important point here. This gametocyte is sexual form and the shape is crescent or banana shape. Okay, here two types of gametocytes are there. One is macrogametocytes, another one is microgametocytes. Macrogametocytes also called female gametocyte. Microgametocytes also called male gametocyte. See, microgametocytes in all the species are smaller. This point is very important. microgametocyte or male gametocytes in all the species are smaller in size, lesser in number compared to female gametocytes. And coming to their staining properties, their cytoplasm stains pale blue and nucleus is larger and stained dark red, and it is diffusible here nucleus. Coming to macrogametocytes, these are larger compared to male gametocytes and numerous in number. Their cytoplasm stains deep blue, nucleus is small, red and compact. Okay, this is the these are the major difference between micro and macro. So see, micro or male. So name itself indicate micro means small. Okay, smaller in size. Macro means big. Okay, so macrogametocytes are larger in size. Okay, next, the prepatent period of this falciparum is 10 to 12 days. So see, these are the gametocytes, also called crescents of plasmodium falciparum. See, male and female, female larger, male smaller. Okay, these are the two morphological forms of Plasmodium falciparum. Okay, one is propozoid ring stage, second one is sexual form gametocyte. Next, coming to Plasmodium vivax propozoid ring stage. Okay, this asexual form ring form occupies 1/3 of RBC. In case of Plasmodium falciparum, it will occupy 1/6 of RBC. This occupy 1/3 of RBC in case of vivax. These are the major differentiation point between ring stage of propozoid of Plasmodium falciparum and Plasmodium vivax. Okay, this Plasmodium vivax can infect only young erythrocytes. Okay, Plasmodium falciparum affect or infect all stages of all age groups of erythrocytes, but here it infect only young erythrocytes. Okay, here one point, pre-patent period definition is the time interval between the entry of the parasite into man and demonstration of the parasites in the peripheral blood is called as pre-patent period. This is 8 days for vivax, for 10 to 12 days for falciparum. See, these are the tropozoite of Plasmodium vivax. See, here we can see only single ring per one RBC. We can see multiple rings in case of Plasmodium falciparum. Next, gametocyte. Gametocyte of Plasmodium vivax. It is round in shape. In case of Plasmodium falciparum, the shape is crescentic or banana shape. But here for vivax, round shaped gametocytes. Okay, here gametocytes prepatent period is for vivax is 4 to 5 days. See the gametocytes. See, these are same features, micro macro, like falciparum. Micro male gametocytes also called microgametocytes, smaller in size. Female gametocytes also called macro, larger in size. Okay, this stains light blue, female gametocytes stain dark blue. We already discussed. So these are the few important points about morphological forms of Plasmodium species. Okay, all of you open your record, page number 119, 119. Open your record, page number 119. In that, parasitology slides and spotters heading is there. First spotter, ring stage of Plasmodium vivax. Ring stage of Plasmodium vivax. Okay, note down the points. These spotters, these charts, very important. You have in your exam syllabus. Okay, note down the points. You have to draw a diagram within that box. Leave that box. Note down the points. Propozoid or signet ring stage of Plasmodium vivax in a thin blood smear. Thin blood smear. So next, you have to note the particular peculiar identification points. Number one, amoeboid appearance. Second one, enlarged infected RBC. Then third point, Scuffner's dots. Scuffner's dots. Next, okay, spotter number two, ring stage of Plasmodium falciparum. See, note down the points. First point, blood smear, peripheral blood smear, showing ring stage of Plasmodium falciparum. Next point, multiple ring forms are seen in RBC. Seen in RBC. Next point, cytoplasm is clear and blue in color. Cytoplasm is clear and blue in color, comma, often with two nuclei, comma, which are stained red in color. Sorry, which are stained red in color. Okay. Next, third spotter, there is one heading Plasmodium. Put heading Plasmodium falciparum gametocytes. Plasmodium falciparum gametocytes. Note down the points. Sexual form, sickle shaped, comma, so called crescents, sickle shaped, so called crescents. Next point, gametocytes. Gametocytes occurs inside the bone marrow and spleen. Next point, cytoplasm stains light blue. Nucleus is scattered in the fine granules. Nucleus is scattered in the fine granules. Next point, pigments are scattered throughout the cytoplasm. Throughout the cytoplasm. Next, in next page, page number 122. After seventh slide, that is Trichinella spiralis. Next, you put heading eighth one, Plasmodium vivax gametocytes. Note down the point, round in shape, round in shape. Next point, male gametocyte or microgametocyte. Male gametocyte or microgametocyte. In that, smaller than female gametocyte, smaller than female gametocyte, full stop. Cytoplasm stains light blue, full stop. Nucleus is large and diffuse. Next point, female gametocyte or macrogametocyte. Female or macrogametocyte stains deep blue, comma, nucleus is small and compact. Small and compact. Okay, leave some space there only. You have to draw these two diagrams. Okay, this is about parasitology slides 2. Any doubts? Okay, thank you.